DNA Replication General Principles. We welcome our listeners today to take a closer look at one of the processes that DNA, the molecule of life, uses to ensure that life is enabled. There are three such microprocesses. They are replication, transcription, and translation. Today, we will take up for discussion the process of replication. Replication is the means by which an organism passes on its genetic information to daughter cells and from one generation to another by making copies of the DNA molecule. It can be defined as the process by which copies of the DNA molecule are made. One may therefore be tempted to say that replication is a process in which Xeroxes of DNA are made. However, unlike Xeroxing, which is an imaging or photographic process, replication is an intricate process in which the original DNA to be copied serves as a guide, the word used is template, and the new molecule is pieced together in such a way that the final product DNA has a new daughter strand and an old parental strand. Such a way of replication is referred to as semi-conservative replication. Our objective, however, is to go into the mechanism of replication and its machinery. In order to simplify our discussion, it will be useful to remember that all the three microprocesses we mentioned earlier have three sub-steps in common. These are initiation, elongation, and termination. By understanding how each of these steps takes place in replication, we will end up getting an overall picture of how replication is achieved and copies of DNA are made. Keep in mind that each new generation requires a complete set of DNA in order to be viable. Before we get down to the nitty-gritty of tackling each of the sub-steps alluded to earlier, we need to turn our attention to an important practical problem that is encountered when replication occurs. You will recall that as discovered by Watson and Crick, DNA is a helical molecule made of two anti-parallel strands running opposite to each other 5' to 3'. In fact, in their famous paper in Nature in 1953, they did mention that the very structure of the molecule gave a clue as to its replicability, as the two strands involved complementary base pairing. Hence, the idea that each strand could serve as a template was proposed by them first. The question that we can ask here is therefore, does the synthesis of the new strands proceed in the 5' to 3' direction, or the 3' to 5' direction, or both? Experimental evidence clearly suggested that the enzyme involved in the polymerizing activity adds bases in the 5' to 3' direction. The chemical reaction that occurs in replication can be set down as follows. DNA having N residues will accept a DNTP and will increase one molecule to DNA N+1 residues, releasing a pyrophosphate molecule. As is apparent from the chemical reaction, in order for replication to occur, a pool of activated deoxyribonucleotide triphosphates is required. A 3' hydroxyl reacts with a free triphosphate deoxyribonucleotide to form a phosphodiester bond with the release of a pyrophosphate molecule. The subsequent hydrolysis of this pyrophosphate in turn drives the reaction in the forward direction. The reaction is catalyzed by an enzyme. Earlier, it was thought that DNA polymerase I, which was discovered first in Arthur Kornberg's laboratory, was the main enzyme involved in the polymerizing activity of replication. It was later found that this was not the case and that the main enzyme is in fact DNA polymerase III. These polymerizing enzymes were discovered in E. coli and named in the succession in which they were discovered. Till today, 3 more such enzymes have been discovered in E. coli, and so the E. coli list of DNA polymerizing enzymes contains a set of 5. The most important of these is DNA pol 3, and it has the following properties. It synthesizes DNA according to the reaction that we have already described earlier. In selecting the nucleotides to be added to the new DNA strand, it is directed by a template. Synthesis is in the 5' to 3' direction only. A primer is required for it to begin putting nucleotides together for the new strand. The primer is usually an oligonucleotides of RNA about 10 to 12 bases long. The enzyme possesses 3' to 5' exonuclease activity, and hence it can remove nucleotides in this direction to do proofreading so that fidelity in copying the template is achieved. Hence, accurate copying of DNA to ensure faithful transmission from one generation to another takes place. Some other features of the enzyme worth keeping in mind are the following. The enzyme is a multi-subunit assembly. It has high processivity, catalytic potency, fidelity, and polymerizing rates of 250 to 1000 base pairs per second. It is made up of at least 8 kinds of polypeptide chains whose designations and molecular weights are the alpha chain 140 kilodaltons, epsilon 27 kilodaltons, theta 10 kilodaltons, beta 37 kilodaltons, tau 78 kilodaltons, gamma 52 kilodaltons, delta 32 kilodaltons, delta prime 32 kilodaltons again. The holoenzyme has a mass of 800 kilodaltons. The roles of some of the subunits of the holoenzyme are worth noting at this juncture. The catalytic activity resides in the alpha subunit and the 3' to 5' exonuclease activity in the epsilon subunit. The alpha, epsilon and theta subunits form a core that is catalytically active but not processive. The beta subunit contributes to the high degree of processivity by clamping the core of the enzyme onto its template. Processivity refers to the capacity of the enzyme to do continuous synthesis without interruption or falling off the template. In fact, scientists who began work on trying to understand replication found that it proceeded in the 5' to 3' direction on both strands of the parental duplex, and that synthesis on both the strands was taken care of by a single enzyme at each replicating segment. This posed an important practical problem, especially since DNA pol 3 synthesizes only in the 5' to 3' direction. We have to solve this problem before we go on. The following figure of a replicating fork, this is what the structure at the replicating point looks like, hence the name, will assist us well in our understanding. You will notice that the 2 parental strands in the figure are separated and form a structure that we refer to as a replication fork. Polymerase 3 synthesizes new strands using the 2 separated strands at the replication fork as templates. The enzyme covalently binds to a new base through a phosphodiester bond to the previous base in the new strand. The choice of the new base is determined through the AT-TA and GC-CG complementarity discovered by Watson and Crick in their structure of DNA. Now you will remember that DNA pol 3 can synthesize only in the 5' to 3' direction. Further, only one DNA pol 3 is at work at a replication fork. As one sees in the figure, the orientation of one parental strand is in the 5' to 3' direction and produces a continuous strand referred to as the leading strand. The other parental strand, which has a 3' to 5' orientation, on the other hand, produces a discontinuous new strand referred to as the lagging strand. The discontinuous segments in this lagging strand are referred to as Okazaki fragments. Reiji Okazaki discovered these fragments, which are about 1,000 nucleotides long. Each fragment has a primer segment at the 5' end made up of ribonucleotides, just like the leading strand. The Okazaki fragments are joined together before replication is complete. DNA pole 1 plays an important role here in removing the primer and replacing it with deoxyribonucleotides. Since this enzyme has this significant role in replication, let us take a look at its properties, especially because its claim to fame is that it was the first template-driven enzyme to be discovered. Here they are. DNA pole 1 is a 103 kilodalton single-chain enzyme. It catalyzes the step-by-step addition of DNTPs, deoxyribonucleotides, to the 3' hydroxyl end of a DNA chain. It also uses its exonuclease activity to remove the primer in the leading strand and the different primers in the lagging strand. This is the key role of this enzyme in replication. It needs the following for synthesis: all 4 deoxyribonucleotides, DATP, DCTP, DTTP and DGTP. Magnesium divalent. DNA template containing single-stranded region is essential. A primer chain with a 3' hydroxyl is needed. It is a moderately processive enzyme and catalyzes multiple polymerization steps, up to 20, before falling up. This, it can be said to have a processivity of approximately 20. It also has 3' to 5' proofreading ability, that is, it has exonuclease activity 3' to 5'. This allows for taking care of editing during polymerization. In general, however, one may note that this enzyme removes mismatched bases at the primer terminus before proceeding with polymerization. In effect, what all this means is that fidelity in the copying process is safeguarded. This is required since only identical copies of DNA will ensure that traits of a species are passed on accurately from one generation to the next generation. It also has an error-correcting 5' to 3' exonuclease activity. It is important for the removal of the RNA primer. It also complements the 3' to 5' exonuclease activity in error correction activities like excising pyrimidine dimers. Let us return to the replication fork and the Okazaki fragments. From our discussion, we are now aware that the overall progress of the replication fork in the 3' to 5' direction is achieved by the ability of the enzyme to polymerize one strand continuously and the other in short segments. The diagram that you see will enable you to get a graphic understanding of the situation. In the diagrammatic illustration, you can see how the DNA pole 3 achieves this by a looping of the template for the lagging strand. When this happens, the lagging strand template is able to pass through the active site of one subunit of a dimeric DNA pole 3 in the same direction as the leading strand. DNA pole 3 has evolved a mechanism so that once an Okazaki fragment is completed, the lagging strand is released and a loop for the next Okazaki fragment is introduced to it. For every new loop introduced to the enzyme, a new short stretch of RNA primer to initiate the formation of yet another Okazaki fragment is needed. It may be mentioned here that this role of forming the primer is fulfilled by an enzyme called primase. The gaps between fragments of the lagging strand being formed are filled by DNA pole 1, which also uses its 5' to 3' exonuclease activity to remove the ribonucleotide primer after it has served its purpose. Finally, DNA ligase joins the fragments. The 4 enzymes that we have talked about so far are DNA pol 3, primase, DNA pol 1 and ligase. One would be justified in claiming that they are the main heroes of replication. The 4 of them, through each of their unique activities, ensures that the elongation step of replication is taken care of. However, before elongation happens, initiation of replication has to take place. How does this happen is the question we will now take up. We can ask the question, does replication begin randomly anywhere on the DNA molecule? The answer is a definitive no. A site has been identified which has been named the ORI, short for origin, site, which is involved in the initiation of replication. The ORI C site in the E. coli chromosome is a sequence of 245 base pairs and has unusual features. It contains a tandem array of 3 13-mers with nearly identical sequences. Each begins with a GATC, a sequence that appears 11 times in the ORI site sequence, many times more often than would be expected on a random basis. The binding of DNA A protein, named for its gene, to 4 sites on the ORI C, shown in the figure, sets in motion the steps for initiation of replication, leading to the unwinding of the template DNA and the synthesis of a primer. Note that the DNA has to be negatively supercoiled to enable DNA to bind. A complex of the DNA B and DNA C proteins joins DNA A to bend and open the double helix. DNA B is a helicase and catalyzes ATP-driven unwinding of the double helix. The unwound part of the DNA is stabilized by single-strand binding protein. It is a tetramer of 19 kilodalton subunits which binds cooperatively to single-stranded DNA. The unwinding of DNA at the origin can lead to positive supercoiling of the circular DNA if the linking number remains constant, and thus unwinding would soon stop. This is prevented by the compensatory action of DNA gyrase, which introduces negative supercoils as it hydrolyzes ATP. The DNA has been unwound, but for synthesis to start, a primer is required. In fact, it was found that RNA primers are made by a RNA polymerase called primase. A unique primer is made for the leading strand, and the first of the many primers for the first Okazaki fragment of the lagging strand is laid down. The stage is now set for the elongation sub-step we mentioned earlier. With the work of initiation done by helicase, rep protein, DNA binding protein, DNA B protein, primase and several other proteins mentioned earlier that are not shown in the figure, the stage is set for DNA pol 3. It is positioned at the replication fork and begins the leading strand synthesis using the primer synthesized by primase in the initiation substep. The duplex DNA ahead is unwound by helicase, rep protein, driven by ATP, like the DNA B protein at the initiation site. The single-strand binding protein keeps the unwound DNA extended and accessible so that both the strands serve as templates. The processivity of DNA pol 3 enables it to continuously elongate the leading strand, while the DNA gyrase concurrently initiates negative supercoils. This avoids topological crisis. At the same time, the second unit of the DNA pole 3 adds nucleotides to the primer of the first Okazaki fragment using the loop available to it. When this loop is completed, it is unclamped and a new loop is offered to this unit to clamp on to. A primer is once again first laid down at this new loop, and the lagging strand arm of pole 3 elongates this fragment, and so on it goes. In the meantime, DNA pole 1 removes the primer from completed fragments and fills the gaps, and ligase joins the final phosphodiester bonds to link the various completed fragments and the completed leading strand. Synthesis proceeds till a termination region is reached, which contains multiple copies of a 20 base pair sequence referred to as TER. These sequences create a trap that a replication fork can enter but not leave. The sequences also bind a protein called the TUS, terminus utilization substance protein, to form a TUS-TER complex. Only one such complex functions per replication cycle, and it stops the first replication fork that reaches it. The other fork stops when it meets this trapped one. Note, encountering each other is enough to terminate replication, and trapping is not essential. The termination site is at the opposite side of the ORI site. The next step to bring replication to a close is synthesis of the last few bases left and untangling of the interlocked concatenated nascent circular DNAs. Separation is achieved by a topoisomerase 4 enzyme. The new DNA molecules are now ready for delivery to daughter cells. It is important to note that many of the steps in DNA replication require the expenditure of a phosphate to sequentially bind and release reaction partners and to move the apparatus of replication along the template strand. Once replication begins at the ORI site, two replication forks proceed in opposite directions till termination at the opposite TER region. Though replication thus proceeds bidirectionally, synthesis of the strands is unidirectional in the 5' to 3' direction. The multi-subunit assembly of DNA polymerase 3 is the key synthesizing enzyme, and polymerase 1 erases the primer on the leading strand and Okazaki fragments and fills the gaps created with ligase sealing the final phosphodiester bonds between the Okazaki fragments and on the leading strand. There are in effect more than 20 proteins whose interplay is required in the process of replication, though all have not been named here. The ones we have focused give a working understanding of the process of replication, which is our objective. These may not act individually, but are thought to be contained in a multi-protein structure called a replisome. These may be assembled only at the onset of replication and be associated with the specific structure of a replication fork. The prokaryotic machinery that we have looked at helps us to understand eukaryotic replication at its basics. Since eukaryotes have several chromosomes with replicating units 100 to 200 kilobases in size, referred to as replicons, what has been discovered in E. coli can be applied to the replicons in other species to get a handle on replication in them, keeping in mind that specifics with respect to the proteins and enzymes and even ORI and TER sites involved in each species will differ. At least one case is known, that of D. melanogaster, in which origins are not fixed but depend on cell type. Whether chromosomal replications have discrete termini is also a question under investigation. In summary, the process of DNA replication involves a complexity of events which in turn involves a range of proteins and enzymes. One may isolate the following steps in the entire process: temporary separation of the parental strands, stabilization of these in the single-stranded state, initiation and elongation of the leading strand, initiation, elongation and linking of Okazaki fragments, joining or termination reactions to complete replication. We hope that these 5 steps are clear to you now that you have completed this lesson.